everyone. My name is Evan. I'm a part of Senior Design Team. The other members of my team are Katriana, Caleb, and Adriana, and our advisor is Dr. Hoshino. Our project is titled An In Vitro Model for Traumatic Brain Injury. Our team is tasked with continuing the development on a novel 3D in vitro model to simulate and study traumatic brain injury. Throughout the year, we have improved multiple aspects of the device design as well as cell culturing and staining procedures used by previous senior design groups. These tests were essential to our design process as they allowed us to identify necessary improvements and conduct preliminary testing without investing significant portions of the budget. Our team was able to make significant improvements to parts designed by groups in previous years. We redesigned components with the hammer to reduce the required amount of pieces to be 3D printed and selected a new material that is autoclavable, allowing for the device to maintain a sterile environment. Adjusted and calibrated spring force to create a more physiological, relevant deformation in the scaffolds, and designed a release mechanism that ensures the waste stack will be released from a consistent position each trial, minimizing variation.
The primary purpose of Trial 1 was to familiarize ourselves with procedures developed by last year's senior design team and begin identifying what needed improvement. When observing the impact images, we determined that the scaffold was compressed far too much and focused some of our design improvements on decreasing deformation.
Based on issues we identified during Trial 1, we shortened the length of the hammer so that in the resting position the hammer would not compress the scaffold as seen in the top image. Also, to eliminate sources of error that come with separating cortical cells from tissue on our own, we purchased already separated cells. The 2D cell plating protocol was also updated to include adding a thin coat of collagen to the dish before cell addition to promote axon growth, as no axon growth was seen during Trial 1. Finally, the seeding density as well as the frequency and volume of media changes were adjusted. On day 1 of Trial 2, we can see in Figure A that short axon projections began to form in our 2D cultures. If you recall the image of our first cell culture trial on day 1 from the previous slide, no axon projections were present. On day 3 in Figure B, we can see robust axon growth and connectivity. However, by day 6, significant cell aggregation had occurred.
Force data for the second cell culture showed the force peaked at just above 15 newtons, much less than the 30 newtons observed during Trial 1. This difference in force is attributed to the changes with the hammer arm. As seen in Figure B, the scaffold in this trial was also deformed past a normal physiological range, indicating impact force needed to be significantly reduced. In the third trial, we compared collagen and PDL as coating materials, reduced deformation of the scaffold by adding cell media during impact testing and adjusting the springs, increased cell survival, and optimized the growth conditions. Additionally, we were able to properly gel the collagen center of the scaffold, as shown comparing image A and B. Most importantly, from Trial 3, we were able to see a significantly reduced compression of the scaffold. The force data, along with the impact images, show a greatly reduced force, which can be attributed to the changes made with the springs and the addition of cell media in the petri dish during impact testing.
In this trial, we compared two different coatings for our standard 2D cultures. Two parameters were used to evaluate neural cell preferences, cell migration, and axon growth. Neural cells aggregate into clumps when they are unable to attach to the cell culture surface. They produce axons only under optimal cell culture conditions. Cells seeded on PDL, images C and D on the right, demonstrate significantly better cellular attachment compared to collagen cultures, images A and B on the left. Comparing images B and D, robust axon growth is seen in cells seeded on PDL, shown by the red arrow, but not in those seeded on collagen. The results indicate PDL is preferred by our cells. Q2 is a live cell fluorogenic nuclear stain that fluoresces after caspase 37 is cleaved in the early stages of apoptosis. Applied to our 2D scaffolds, it will allow us to monitor apoptotic death over multiple time points in response to induced traumatic injury. Here, we validated its function in 2D cultures of our cells. Apoptosis was induced by oxidative stress using H2O2. On the left, images A and B, you can see cells stained with Nu-Q after induction of apoptosis, and on the right, a non-apoptotic control group stained with Nu-Q. As expected, the apoptotic groups had significantly higher levels of apoptosis, indicated by the increased fluorescence compared to the control group. This validated that Nu-Q is functioning in these cells. Scaffolds were stained with immunofluorescent antibodies related to tubulin 3, a component of the cytoskeleton that is unique to neural cells. On the right, Figure 16 shows the 3D projections of confocal images obtained from the scaffold on the left. Cells are clearly visible, validating that neural cells were successfully cultured on our scaffold.
After testing, it was seen that the cover required too much assembly and disassembly of the device before and after testing, as a design change would be recommended. Adjusting the spring force and having potential variations for a variety of impact forces would be a marketable selling point. And finally, having justification of how in vitro 3D models can be used through simulated methods would validate the results gathered.
We would like to thank all of those listed above for their help during the course of this project.